Good morning. My name is Samantha Riles, Communications Officer from Sugar Research Australia, and I'd like to welcome you to today's webinar. Gerard Scalia will be presenting on yellow canopy syndrome. Please note that today's meeting is being recorded and will be available on our website. At any time that you are not speaking, can I please ask you to remain on mute to keep the background noise to a minimum? If you are having any technology issues, please send a message in the chat box, which is located on the menu bar of your screen. I will be assisting as a co-host to ensure all runs smoothly. At the end of Gerard's presentation, he will be taking questions, and we encourage you to take an active part. You can type questions into the chat box throughout the presentation, and I will moderate these at the end. At the end of the presentation, if you have questions, you can also unmute your microphone and ask Gerard. I'd now like to hand over to SRA's Central Region Adoption Coordinator, Phil Ross, to introduce our speaker.
Thanks, Sam. Yes, Sugar Research Australia is running this session this morning, mainly because the 2020 grower updates were cancelled due to the COVID-19 issue. And we know that yellow canopy syndrome is of high interest to growers and advisors alike. So we thought we'd take this opportunity to put it out there in webinar format. Our speaker this morning, Gerard, Gerard is a molecular geneticist and has been with SRA and YCS for 11 years. And he's been working in the integrated YCS program since 2014. So he's well over the top of all aspects of the program. So I'll just hand over to Gerard now to run through the program. Thanks, Gerard.
Thanks very much, Phil and Sam, and good morning, everyone, and thank you very much for joining with me for this webinar today. The presentation I'm going to show you is going to cover off on the YCS research that has been conducted since 2014, and hopefully it will give you a better understanding of what YCS is and what causes it. Now, I've got a lot to get through, so hang on to your seats. So the outline of today's presentation: frequently asked questions and answers, symptoms and identification, the key findings, what is the cause of YCS, the trials that are currently being conducted, and summing up and conclusions. So what is YCS? YCS really just describes what you see. It's the condition that describes yellowing in the mid-canopy of sugarcane. It was first seen and noted in Gordonvale in 2012, and since it's been recognised and confirmed as far south as Maryborough in 2016. The time of the year that you see it most, the YCS season, is the peak growing season. So usually it's during or between December and March. However, I will stress at this time that you can see YCS at any time of the year if the conditions are right for its development. But that period between December and March is the key time.
Plus 61747221976 is now joining.
So where do we normally see YCS? Which leaves are normally affected? It's the leaves of the mid-canopy. Leaves numbers 3 to 6 are the main ones. So just so that we can get the numbering right for the rest of the presentation and they're all on the same page, if we look at the stalk, and for those people who aren't aware of how the numbering is of leaves goes, if you identify this hinge area between the leaf blade and the sheath, that's called the dewlap. And if you go up the stalk and find the first one that's first leaf that's fully expanded, you'll be able to see that dewlap, and it's like an orangey brown ring around there. That's the first visible dewlap, or the top visible dewlap, and that first fully expanded leaf is leaf number 1. And the one underneath is leaf number 2 and number 3 and 4 and so forth down the stalk. This is where YCS normally starts. So how do we identify YCS? It can be a little tricky, and to be confident, you have to have all of the following conditions met for you to be confident that YCS is in the field. You have to have yellowing in the mid-canopy as explained. The symptoms have to have a particular pattern of development, and they have to have a particular colour. It's a more golden yellow colour, which I'll speak to in shortly. Leaves have very high sucrose content, and they also have an abundance of starch in the leaf at first light, which you can stain for to make it more obvious and visible. So it's difficult to identify in the field because, as you know, sugarcane has lots and lots of forms of yellow. Yellow caused by herbicides, yellow caused by water stress, yellow caused by nutrient deficiencies. So it does make it difficult to confirm. So we tried to come up with a diagnostic. We identified 6 genes that seem to be unique to YCS. And if you have a look over in the top corner here, this gel, it shows where we tested some YCS samples and some control healthy samples, and we got a hit with one of these genes. And here we didn't. So it's a little bit like them testing now for the coronavirus, a very similar method using PCR. So then we thought, well, that's pretty good. Let's see if we can test this against all the other forms of yellow out there in the industry that you see in sugarcane. And unfortunately, when we did that, we actually got a hit on some of the other yellows. So it wasn't, and we tested all 6 genes and it fell away. So it wasn't a test that was unique for YCS, so we couldn't take that research any further. However, I think that it tells us something about the cause. The reason this failed, it might tell us a lot about the cause of YCS, and I'll come to that soon. A diagnostic tool, however, has been developed, and it is being validated as I speak throughout the industry, and has been out for a couple of years, and that's the midrib stain test, and I'm going to talk to that now. We needed to have something that we could help reduce the misdiagnosis, something that someone from Prod Services or someone from one of the researchers could take to the field. It had to be user-friendly, it had to be quick and easy, it had to be safe to use, and it had to give you a fairly high accuracy rate. And it also was, we were hoping that it could help predict the possible onset of YCS. And the kit contains things like a stain, a hand lens, and some flashcards. The first flashcard asks you to make sure that you're looking at the right part of the canopy. Not in the upper canopy, where everything is totally green. Not down here in the lower canopy where all the leaves are dying or senescing, but this mid-canopy between leaf 3 and 6. And the trick here is to pick up, to be confident in confirming YCS is to pick it up early. If you see the yellowing occurring, just starting to occur in this region, say on a leaf 3 or a leaf 4, then leaf 5 and 6 underneath it will be green. Leaf 2 and 1 and all the upper canopy above it will be green. So you'll have a green zone above it and a green zone below it. However, if you don't pick it up early, it'll start to get really, really yellow and start to die. And eventually, as this crop grows up from the top up, all the dead leaf, this leaf will become a lower leaf and it'll die, and all the brown leaves will join up with the yellow. And so it'll be very difficult at that point to say this crop has YCS. So the secret is picking it up early. So what do the symptoms look like on the leaf? Top leaf is a healthy asymptomatic control leaf. When YCS starts, you normally get some yellowing on one side of the midrib. Here's our midrib. And that usually starts around the middle, but it doesn't always have to. It usually starts where there's the most sun hitting the leaf. Now, as most leaves bend in the middle, that's where most of the sun hits the leaf, and that's generally where you see the yellowing start, because that's where most of the sucrose is being produced. However, it could start further up here or further down here, depending on the geographical orientation. We also note that you see a lot more YCS on the outer rows of a field where it's getting more sunlight. So it starts close to the middle, a little bit yellowing occurring, and then it starts to progress and it moves towards the tip and towards the base, and then it starts close to the midrib and moves out towards the margin. Now, I suggest not looking at leaves that have yellowing from the tip that go down the margins, down the outside, as being YCS leaves, because that's more akin with natural senescence or sometimes with a nutrient deficiency. As it progresses, you see it becoming more and more yellow, and it normally affects one complete side of the leaf, one side of the midrib. When it becomes more advanced, it progresses to both sides, and then you get this beautiful colouring that really starts to exhibit, and I'll just zoom in on it. And you can see it's a really golden yellow colour, and it can be quite blotchy. It doesn't have to be completely uniform, as you see here. And that golden yellow colour is because the underlying pigments, the carotenoids and the anthocyanins, are coming through to the surface when the green pigment is being lost. That's very much like what you see now in autumn with the autumn leaves, say your beautiful maple leaves, where those other colours come through when the green pigment dissipates. So that's the colouring and the pattern of development that's unique to YCS. Now, we noted that the leaves have a lot of starch in them because when they get too much sugar, they start to store carbon in another form, and starch is a real energy form. It can store it, and it naturally does this anyway because it needs energy at nighttime. So it starts to put carbon into starch to try and reduce the amount of sucrose that's being produced. Annalie, who was working with me at that time, she realised that we see a lot of starch in the midrib, and while it's high in the leaf blade, it's hard to see it in the thin blade, but you can easily see it here in the veins of the midrib when you stain it up. So you just add some stain, cut your leaf across, add some stain to the midrib, and leave it for about 20 seconds, and the starch will stain up, as you can see here. And in a healthy control leaf, you don't get any staining up. But if you only use this stain method to check for YCS, you'll get a lot of false positives. And I want to stress that point, because if you do this on a water-stressed leaf, these will stain up. So you need to marry this up, this staining, with all the other things that I've previously just shown you. You have to be in the right part of the canopy. You have to have the right pattern of development. You have to marry all of those things up to be confident. And when Mike Turner and Veronica Chapman in Bundaberg tested the kit for us a year ago, they got an 86% accuracy rate by doing that, because we ground truthed everything by measuring actual sucrose and starch in the leaf in the laboratory to go with their diagnosis. So it was a pretty good accuracy rate when you put everything together. So what are some of the key findings from the YCS research? And there are many, and I can't discuss them all, but these are the main ones. YCS is always preceded by some form of stress. That's a given. It could be abiotic or biotic, that is non-living stress or a living type of stress. So water stress, too little, too much, heat, oxidative. Could be nutrient stress. It could be a biotic stress like some pest pressure, insect pressure. We also note that the leaves have very high, YCS leaves have very high silicon content. And if you, a lot of literature out there that would suggest that high silicon uptake into a leaf is it strongly correlates with abiotic stress. So I think that's interesting. There's another sign that, you know, these plants and these leaves are under stress. YCS usually expresses after good rain. Now, we get a lot of phone calls from growers who say, look, it's rain, about a week later, get a flush of yellow going through the field. So what is that about? And that's a very common thing. If you've had a really dry period or some form of stress period that slowed the crop down, and then it starts to rain, and you get plenty of hot, sunny days that follow for the next week or two, then of course you're going to get a really rapid growth. And That rapid growth leads to yellow. And that's what I want to focus on now in the next part of the presentation. So we believe that growth rate drives YCS development and not vice versa. A lot of growers call me and say, look, YCS has caused my crop to slow down. Well, I would argue the reverse. The crop has slowed down, and as a result of that, YCS exhibits. So if it's all about growth rate, what can or how can you measure the growth rate quickly in your crop? How can you get an idea of whether there's a problem? And a really simple way to do this is to simply just go out and measure the lengths or have a look at the lengths of the internodes. And by the internodes, I mean, so if we look at the stalk, that region, there's a node, there's another node, that's where the bud is and where the leaf shoots from. The distance between here, the region between here and here is the internode. If you look at the length, it'll give you a good idea of growth. But if you also look at the girth and look at its overall volume, that will give you an idea of the growth rate. So it's a very simple thing to do, and you can learn a lot from that just by simply going out and cutting some stalks and stripping them back. So if YCS is all about growth rate, we needed to put our money where our mouth was. That was our hypothesis, that slow growth rate was driving YCS development. So back in 2015-16, Davey Olsen conducted a growth regulator trial in the Burdekin, where we attempted to induce YCS artificially. We said, well, let's slow it down on purpose. And the way he did that was to use a chemical called Moddus, which you all know is a ripener. And that ripener you put onto your crop, before you want harvest to improve the CCS. But the thing about Moddus is that it's a chemical that stops a hormone called gibberellic acid. And it's a GA inhibitor, and GA causes elongation of the internodes. It causes them to grow long. So if you inhibit it, the internodes will only be short. So this is our way of reducing the growth rate. And he did this and applied it, and in the field, he called us and said, look, there's yellowing. So we went and had a look and to take samples. And there was healthy green stalks in that field. There was also naturally occurring YCS, and there was the Moddus treated plants that also had what looked like YCS. And it happened to be in a leaf 4 where this yellowing was developing. So we went and harvested the stalks and we measured them up. And if we just focus on internode 4, which is the internode beneath the sheath of leaf 4, and you can see the volume of a healthy green stalks, internode 4 is around about 55 mil. And here is the YCS stalk that are naturally occurring in that field, and the Moddus stalk, and it's around about 35. So it's about 40% smaller volume, and we're able to induce YCS in this Moddus plant. And the striking thing is a very similar size internode underneath that symptomatic leaf. So that was a really important experiment that we learned a lot from. So I said before that one of the key things about YCS is that the leaves always have high levels of sucrose. So what causes high levels of sucrose in the leaf? Well, here's your leaf. Here's your stalk. Said before that those leaves 3, 4, 5, 6 are your powerhouse leaves. They're the ones doing most of the photosynthesizing. They're the ones that are producing all the sugar. So they're your source leaves. This is your factory, so to speak. Over here where they've got to send it to the internodes or to the roots, that's your sugar sink. That's where the sugar's got to get to. That's your warehouse. So it's like running water downhill. The steeper that hill, the faster that water will be. The steeper the gradient, the faster it will flow. For sugar to leave that leaf and get to the stalk where we want it to be, because that's what we want it to do, we need to have a strong sugar gradient. It has to be from a high concentration to a low concentration. If you do that, sugar will flow readily and you won't get accumulation here. So sucrose accumulation in the leaf can be caused by any factor that slows down this transport, that reduces this sugar gradient between the sugar source and the sugar sink, between the factory and the warehouse. What things can do that? Well, small internodes, like I just shown you in the growth regulator trial, can do that because your sugar sink size here is small. Small root system could also do that. However, Anne Ray from CSIRO conducted a fantastic research on YCS root system compared to healthy controls. And after that year of research, she concluded that there was no difference, that she found no difference between YCS root systems and healthy control root systems. So that would suggest that limitation in the sink size wasn't in the root, but in the stalk itself. However, you know, while I don't have data, I suggest that if you do have a small root size, that could cause serious problems as well. The other thing which you would have already thought of just now by looking at this is that the other way that sucrose could accumulate here in the leaf is if you disrupt this transport system. So if you have some form of physical blockage in the leaf that stops the sugar from getting out and getting to the stalk, and we'll talk about that as well today. Okay, so let's look at this transport system and the sugar gradient. Here's your leaf, okay, and the stalk underneath it and the internodes. Now, as a general rule of thumb, these leaves, when they're photosynthesizing and producing lots of sucrose, about 80% of the sugar goes down to the two internodes beneath it. Okay, there's the leaf sheath, there's the two internodes beneath it, about 80%. The other, and some goes down to the roots, of course, because that's another big sink. The other 20% goes upstairs here to the canopy, the upper canopy, to the leaves above true leaf number 1 that I showed you before. And those are sink leaves as well, and they need to grow, and they don't export any sugar. They need to get some sugar to help them grow. And so 20% goes upstairs, but 80% goes here, the lion's share goes here. So if you have a large sugar sink, large internodes, a large warehouse, you have a really fantastic sugar gradient. Sugar leaves that leaf very easily and gets used down here for growth and for storage. You'll get low sucrose accumulating in the leaf in this situation. However, if you have the opposite to that, if you have a small internodes here, you have a small sugar sink, at first it'll be fine. Sugar will leave the leaf and it'll start to come here and there'll be a strong gradient and it'll move really rapidly. But as these internodes fill up, that will slow down and potentially to a trickle. And so you will have a reduction in the sugar gradient and eventually sucrose will build up in the leaf and it'll become yellow. Just want to make one more point here as well, is that the interesting thing is that this leaf was green before as well, and that if you measure the sizes of green healthy leaves and YCS leaves, they're the same. There's no difference in the size of that factory, and that makes sense. It's just when you change the size of the sink or the warehouse that you cause problems in that factory. Sorry, I'll just go back there. And that point is very important. The length of these internodes here are determined by a few things. They're determined by the availability of water and nutrients, but they're also determined by what we call degree days or heat units. And they will elongate for a set period of time and then they will stop. And in our commercial crops in Queensland, it's around about 380, 400 degree days. After that time, they won't grow any longer, no matter what you do. So it's really important to grow the biggest internode during that period of time that can be grown to get the biggest sugar sink to reduce the risk of YCS, to reduce this risk of sucrose accumulating in the leaf. Now I got called to a field and it had YCS there, and I went in and I cut 3 stalks. Okay, the one on the right was healthy and didn't have any YCS. The other 2 had some YCS, and the one in the middle was the worst, even though it's different in length. And I encourage you all to go and do this. If you've got what you think is YCS in a field, to go out and cut some stalks where it's looking nice and healthy and where it's yellow, and strip off the leaves and number them as I showed you before. So this internode number 3 is beneath leaf 3, and label them up and put them side by side and have a look. We know this. Remember, this crop grows from the top, not from the bottom, and so it puts on a new leaf and a new internode approximately every 7 to 10 days in summer. So this internode 3 and this internode 3 and this internode 3, they're all the same age. 4 and 4 are the same age, 5 and 5 are the same age, all formed at exactly the same time. The lengths, however, dependent upon what's happening, water, nutrition, and so forth. And so if a crop is under stress, you'll see a reduction in growth. And this is the point. And you can find out you can learn a lot. Like this is like looking at growth rings in a tree. It shows you growth rate. And if you look at this stalk, internode 3 down to internode 12, there's 12 or 9 internodes in that distance there. This is 3 to 9. So these internodes, look at the size of that internode 4 there compared to that internode 4. This is a much larger sugar sink. It's also thicker. So if you did the whole overall volume, it would be an enormous compared to this one. So this has a capacity to accept way more sugar than this can, and this will have less risk of sucrose accumulation in the leaf because it'll be able to accommodate everything that the leaf is producing. And so you won't see yellowing develop. You can also work backwards and find out, okay, well, if this is once every 7 to 10 days, you can go back and say, can I identify a period of time where something might have happened in the field that caused extra stress to slow the crop down? And if you can identify that, then maybe you can alleviate that issue and help the crop to get going again. So the other thing I mentioned was that, yes, you can have a small sink size or small internodes or slow growth, but what about a physical blockage? What evidence is there? Which one is causing sucrose to build up in the leaf? Is it slow growth rate or is it a physical blockage in the leaf? Okay, so if it's a physical blockage, if it's a complete blockage or is it a partial blockage, it'll slow this sugar getting to the internodes. Well, look, a very easy experiment to do, we thought, was to take a leaf, okay, and have a look at it, because if you had a complete blockage, a total blockage, let's say here across the middle of the leaf, then no sugar should be able to leave the leaf and get down to here. It'll get to here and stop, and so it should build up here. So this part of the leaf should have a lot more sugar in it than this part of the leaf. So we did that and we tested it and we looked at how much sugar and starch was in there. And let's just focus on the sugar. And here's, just look at the green and the yellow bars here. This is the amount of sugar in that leaf. And we cut the leaf into quarters. This is the tip, the mid-tip, the mid-base, and the base, and also the sheath. And we had a look in there. And what you can see if you follow the green and the healthy leaf, that sugar increases along that leaf steadily towards the base and into the sheath. And in the YCS,
unknown participant is now exiting.
You get exactly the same pattern where you get an increase in the in sugar in the YCS leaf, but it's just a lot higher. So we didn't see accumulation just here or just here. So it was an indicator that, yes, sugar was actually moving along that leaf and getting to the sheath, so that blockage may not be there at all. This is showing you a combination of lamina, and the lamina is the leaf blade, and the midrib combined. But if I separate them out and just say all of the leaf blade, all of the midrib, and all the sheath, you can see the same pattern. So the sugar is made in this part of the leaf, in the blade, and then it sends it into the midrib, and then it comes down to here. So it looks like there's no problem in terms of a physical blockage because the pattern is similar. You might be saying, well, that's all very well, Gerard, because, you know, we know sugar can get to the end of the sheath. So what? What about the stalk? Maybe there's a blockage in there, and then it starts to build up here. So we had a look at what was going on in the stalk, and we had a look at the sugar concentration in these two internodes, and here it is here. And you can see that there's the healthy one, and here's YCS, and it's got as much sugar, if not a little bit more, than in the healthy one. So sugar is getting into the internode in a YCS stalk. We had a look a little further. Here are three main commercial varieties, Q200, KQ228 and Q208 in three regions, Herbert, Burdekin and Mackay. And we measured the amount of sucrose in the internodes, internode 4 and internode 6 in those stalks, in those three varieties, in those three regions. And where you see 2As, statistically that means they're the same. And what looks like you got more here and less there and whatever, in each of these regions, you can see that there's no difference in the sugar content in those internodes between healthy controls in the green and YCS stalks. Take it a step further, we said, well, okay, that's doing our analysis in the lab. What about if we just replicate what's going on in the mill and run it through a juice lab? So in the trials last year in the Burdekin and the Herbert, we did the same with a whole lot of stalk sample from the field, ran it through the juice lab, and these are the CCS results for healthy controls and YCS. There's no YCS impact from YCS, no CCS impact from YCS, sorry. Just remind everyone some of the major things that can affect CCS, and that is things like the plant age, crop class, suckering, lodging, shading, flowering, leaf nutrition, and water, just as a reminder. So why is high sugar in the leaf bad? Why is that such a problem? Well, when it accumulates in the leaf, what it does is it starts to destroy the green pigment, the chlorophyll, the thing that's needed to make the sugar. And that's what makes our leaf look green. And how much loss is there? Well, when you measure the amount of chlorophyll in the leaf, there's about a 40% loss. And that's quite significant. So why does sugar cause the leaf to go yellow? Well, here's a leaf. If we flip it over and look at its underside, we see these pores called the stomata. And these pores allow carbon dioxide to come in for photosynthesis, but they also allow water and oxygen to leave. Now, when you get high sucrose in the leaf, these guard cells here close, and that pore closes off. And when that closes off, CO2 can't get in, yes, but water can't leave. And when water can't leave, the leaf can't cool down. It's a little bit like closing off your sweat glands. And you'll get an increase in leaf temperature. So that's bad. You've got sucrose climbing in the leaf, and you've got leaf temperature increasing. Recipe for disaster. But the plant's very smart, and it's got molecules within it, these signal molecules that act like little sensors, and they can pick up or register how much sucrose is there. And then say, oh, let's try and slow down the sugar production. Let's send a message to the factory in the leaf and tell it to slow down. And by doing that, it down-regulates photosynthesis by turning down the genes that are associated with photosynthesis. So we've measured this, this down-regulation. Unfortunately, when you turn down the genes that regulate the photosynthetic genes, when you turn them down, it's almost like shooting yourself in the foot, because it disrupts the flow of electrons through the photosynthetic machinery. Think of it like this. Think of it like you've got a fuse wire and you're running electricity through it, and then all of a sudden you slow that electricity down by increasing the resistance of that wire. You slow that electricity down, that energy is going to go into a different form. It's going to be transformed into another form, and usually that's into heat. And that wire will heat up, and it'll keep heating up, and as you know, that fuse will eventually burn. And that's pretty much what happens in the leaf. It burns from the inside, and we call that photooxidation. And because this, while it's turned down to photosynthesis in the leaf, it can't turn off the sun. And so energy continues to come into the leaf, and that energy has to go somewhere. And that energy goes off into these other molecules, and it causes a breakdown of membranes. And when it breaks down the membrane of the chloroplasts, which contain the green pigment, it destroys them. And when you destroy the pigment, it goes yellow. And the other carotenoids come through, the other pigments come through, and that's what you see. So we understand very clearly why the leaf goes yellow, and it's all driven by high sucrose in the leaf. So you should be asking yourself, how much sugar is too much? What's too much sugar? What causes it to go yellow? How much? Well, we all have different tolerance levels to certain things, but it doesn't matter what the variety is in our commercial canes, it's all about the same. I've tested thousands of leaves and measured sucrose in them, and what we find is that if you have a concentration higher than about 200 nanomoles per milligram of dry leaf matter, it'll start this process of turning yellow. So here's a green healthy leaf. Just focus on this part, and here is a yellow leaf. Here's your 200 upper threshold. If the sugar doesn't climb above that 200, the leaf will stay green. It won't tip over and become yellow. But if it passes, then you'll see yellow expressing, and that's your YCS leaf. And when this happens, there's no coming back. This is lethal, okay? You can't reverse it. It'll stay yellow and it'll continue to be yellow. So the trick is trying to keep the sucrose underneath that 200 level, and that is stop the accumulation from rising to toxic levels. Okay, in the beginning, a lot of people thought, well, maybe YCS is a disease. It's caused by some sort of disease. Is it caused by a bacteria, by a fungus, by a virus, by a phytoplasma? Is it caused by some loss of beneficial microbes in the soil? So leaf samples from were taken, stalk, roots and soil, and checked. And we used an pathologist check for it, and they even said the epidemiology didn't look like it was a disease. But nonetheless, we used the most sophisticated analyses we could, and next-gen sequencing is that, where we take these samples and look at the genetic sequence of everything except for sugarcane, so for all of these sorts of things, and then we have a targeted approach and we look for specific bacteria or fungi or viruses and see what we find, see what we get a hit on, just like checking for the coronavirus at the moment, or an untargeted approach where we look at all the sequences and we look for groups of functional genes or pathogenicity in a complex and see what we could find. And in both these methods, when you run it through the software analyses for a single pathogen, a disease complex, or a loss of microbes, and we did this for 1,000 samples, more than 1,000 samples over that time period from 14 to 19, and checking for all of these things, and even looked at metabolites and proteins and gene expression and so forth, ran trials with antimicrobials, antivirals and fungicides. Priya Joyce and Russia, who did some fantastic microscopy work looking for these organisms in the leaf and in the stem and in the roots, DNA and RNA sequencing. We threw everything at it, and for many years sent samples overseas, did everything, and still came up with none. We couldn't find something that was consistently present in all leaf samples, in all YCS tissue. We did get a hit occasionally on a bacterium or potentially on a phytoplasm, but it wasn't everywhere, and it wasn't in high titer or concentration to cause a disease. So no evidence in this area. Now for a big one, YCS and yield impact. In some of our trials, we see significant yield impact. Other times, we repeat that trial, we don't see any impact. So it's a bit of a mystery as to how much yield impact YCS can cause. I just want to show you the results of a trial from last year, and we had a series of different types of chemical treatments, but these bars represents tons of cane per hectare, so up around about 110 tonne per hectare on average across here. And this orange line is YCS severity, number of leaves. And I think you can see at a glance that even though in this case, you've got really high yield, you've also got high YCS severity, and here you've got high yield, just as high yield, but low severity. So there's no correlation between the two. I would argue that a better measure of potential yield loss would come from the duration of YCS symptoms. And by that, I mean, take for example, if you've got, say, 5 leaves in your crop that go yellow because of YCS. In the whole crop cycle over a 12-month period or longer, you might have a canopy of 50 leaves. So 5 of those 50 go yellow because of YCS. So 5 out of 50, 10%. But it's not 10% loss because YCS leaves aren't running at a negative the whole time. Okay, so it's running at about, let's say, 40, 50% of the time. So if we say 50% less photosynthesis, then that 10% becomes 5%, and so therefore you may have a 5% loss. Now, I would argue that you could walk from one end of your field to another and see a 5% decrease in yield for many other reasons. So YCS may not be a big issue if it's not there for the long time. If it's there for a lengthy period of time, like say you lose 10 leaves, then yeah, you're going to have some yield loss, there's no doubt about it. But we've got trials that are investigating that at the moment to see if we can get clarity as well on that issue. So that was in field trials and also sampling fields to see what the issue is. But I thought, well, what has YCS done across the entire industry? So these graphs here that I've put together look at the last 10 years. So we've got tons of cane per hectare on this axis, and over here we've got rainfall. And I've done this for each region where YCS has been confirmed. So just to sort this out so that we're all on the same page here, these graphs represent the 3 seasons, spring, summer, and autumn. And this is a total rainfall for each of those seasons, because these are the growing seasons for sugarcane. And of course, when we're dealing with summer, this orange bar, we've got December, but then we've got January and February from the following year. So that's why it's 2010, 2011. And then of course, autumn through to May. And I've plotted the tons of cane per hectare over the top and when YCS appeared. So let's start with Gordonvale. YCS appeared in 2012-13 season. There it is there. And I want to point out as well that 2010 was confirmed by the Bureau that of Meteorology that this was our wettest year on record. And 2011 wasn't far behind. And if you go through these slides, I'm not going to spend a lot of time on this now. It's being recorded, so you can have a look at it, your heart's content later on the website. But you'll see that, yes, these 2 years are quite wet. And when you get extremely wet years like that, you don't get much sun, okay? And you know, you get too much water and you get low yield. And just about in every region, as we go through, you'll see that the yield is lowest in that time. Anyway, YCS appeared here. And in Gordonvale, since YCS appeared in 2012, sort of got, you know, more and more yield each year until 2015-16. Now, you see this beautiful uniform pattern in rainfall when nothing goes wrong. And that's when I say nothing goes wrong. I've put on here for you all the serious weather events that have occurred, where the cyclones have been or where there's been a severe tropical low. And I think this speaks volumes in terms of what affects yield significantly. And we all, it's not rocket science, we all know that these weather events have drastic effects on yield. But here we can see when you get a really good season, one after the other, you get a massive increase in yield. Interestingly, if I talk about the entire state of YCS symptoms, this was the year when it was at its worst, 2016. Now, some of you say, no, we had much worse YCS in 2013. And I agree, you did. Some regions you did. But right across Queensland, 2016, that's where YCS was the worst on a large scale. And that usually equates, 15 and 16 usually equates where we had our highest yield. And as I said before, for YCS to be visible, you need to have that sunlight. Okay? So without it, you don't see really bad expression. And that was true from the very beginning. Back in 2012, 13, you know, people said, Tully doesn't have any YCS. Why is it? It's just near Gordonvale. Why doesn't it have YCS? Well, everyone knows the thing about Tully is so much rain and less sunny days, fewer sunny days. But guess what? When Tully had its highest yield in 2015 and 2016, that's when we started to see some YCS. So if your crop slows down and then you get good rainfall and you got good period and you get lengthy periods of nice, hot, sunny days where you can produce a lot of sugar following a drier period, then what I'm suggesting is that you get high supply, more than what the warehouse or the stalks can accommodate. Supply is greater than demand, and then you'll see a greater potential to see yellowing in the leaf. Doesn't mean it's a bad thing, it's just that there's a higher risk during that time. I'm not going to go through these now, and you can have a look at them, and I think hopefully you will find them quite interesting as to when you think you could be going to get a fantastic yield that year, and then all of a sudden something happens like Cyclone Debbie or Cyclone Yasi, or a severe weather event like we saw in the Burdekin last year in January, or 2 years in a row actually, and huge rainfall again this year. So I think yield, YCS, there are bigger impacts on yield than YCS, I would suggest. And that's the last graph there at Plain Creek that is on this. Oh, sorry, Maryborough, as far south as it's gone. So why 2012? Okay, why did YCS occur in 2012? What was different about 2012? Well, I would suggest this to you. If we look at temperature, I think YCS appearance and distribution in Queensland strongly correlates with abnormal water and temperature profile. So bear with me here for a moment, and you can hear me by listening to my theory here. But if you look at the last 110 years of temperatures, and I put a linear regression line through here, through about 1965, I think you can see a pattern that since 1965, it's got a little bit hotter. And it's only about 1.5 degrees, you might say. Well, so what? That's not much. But it could be significant when you put it together with other things, like severe weather events. Here in 2011, that really wet year, 10 and 11, you can see it's a little bit cooler, and then YCS 12, 13 is where it hit. Look at what's happened in this last period here, these last 9 years or so. It's been significantly hotter. So if you thought it has been, it has been. And you say 1.5 degrees, but you know, that's not much. But you know what, if it's 33 degrees and then all of a sudden it's 34.5 degrees, you'll notice it. And maybe that little bit of extra stress, along with other things, has contributed. So what has the Bureau said? The Bureau said that in the last 20 years or so, we've had an increase in our summers. And in fact, now our summers are 1 month longer and our winters are 3 weeks shorter. And we actually have an extra 10 days in summer where the temperature average is above 34 degrees. That's around about 30 days in total now where we have temperatures above 34. Now, I would suggest that if you don't have access to irrigation and the plants are water stressed, that's going to be an issue in terms of growth rate. And if you look at these colours of the whole of Australia, you can see when YCS appeared in this period here, it certainly got a lot hotter. Now, I didn't produce this map. This is what's on the bomb site. And when you look at that, isn't it interesting that this period here, which is the YCS hot zone between Gordonvale and Mackay, okay, is 1.5 degrees hotter than everywhere else. So a little more stress, I would think, in that region, and adding more stress to the crop and slowing it down this growth rate, potentially leading to more YCS since 2012. And YCS started here and moved down the coast, and it has got progressively hotter as we've come down here. When I was in Maryborough with my team in 2016, we confirmed YCS there in Maryborough region. It was 36 degrees on the 1st of April. That was unheard of for that region. And people in Maryborough who may be listening today would remember it. And so it has got progressively warmer and more stress, and the weather events have been extreme for the last 10 years. I'm not saying they're going to stay that way. It's just what the records show. There's other things that have happened as well. Economic pressures, okay? World price, regulatory pressures in terms of nitrogen. Power, your costs, you know, it's just being exorbitant. You know, electricity price has gone up 130%. Your pumping costs are enormous. Cost of production is huge. Put all these things together, more pressure, more stress, may not always have available resources to keep that crop, to reduce the stress on that crop. On a good note now, crops do grow out of YCS. The best way to manage them is to not let the crop slow up. Now that's easier said than done, but follow best practice when you can to reduce stress on the plant. What is the cause of YCS? The big question. Research doesn't support a single cause. It's anything that slows up the growth rate to the extent that it significantly disrupts the supply-demand imbalance, that factory warehouse imbalance. Environmental or biological stress or both can trigger YCS. Accumulation of sucrose in the leaf above that tolerable level, upper level, is what causes the yellowing. Now look, we believe that insects may be involved because where we've run trials and had a broad spectrum insecticide applied, which took out every single insect, we had a stay green effect. The crop didn't get YCS. Now we took every insect out, okay? So we've been doing these experimental trials with trying to look at the efficacy of a chemical and whether we can learn something about that, and whether it's a particular insect or a group of insects that may or not be involved in YCS development and symptom expression. So this chemical option is under investigation, has been for a year or so now. We have trials in the field. It's not imidacloprid, and I stress it's not registered for sugarcane above ground. And it's really just to confirm or eliminate the role of insects and to help us get a handle on yield. And it's being conducted in 6 locations this year in Queensland. And the outcome of those trials, which will be later in the year when they're finished, will be pivotal to any YCS management program going forward. So the trial in the vertic end is looking at efficacy to suppress YCS symptoms by applying it at different parts of the plant and at different times, either weekly or fortnightly, and at a couple of time points. In Ingham, it's looking at the effect on varieties, where there are 16 varieties in a field, and evaluate varietal susceptibility. So far, while that's been running, we haven't seen any true differences between the varieties, even though I know some people say some varieties are worse than the others in terms of YCS susceptibility. Maybe that's got a little bit more to do with how some varieties hang on to their leaves more than others, and so it looks worse than it actually is, potentially. To investigate efficacy again there, and commercial strip trials on growers' properties. We have 4 of those going, where we're applying the insecticide with commercial boom sprays to see whether we can get any result, and the results of those will be out later in the year. Our head entomologist, Kevin Powell, and his team are collecting insects on sticky traps and sticky tape in the fields, and trying to identify if there's one or more insects that could be the culprit, and in potentially providing more stress or doing something else to bring on YCS symptoms. And we ground truth everything in these trials by taking leaf punches and checking for sucrose and starch along the way. So in conclusion, YCS is not a disease. It's a physiological disorder. Slow growth rate, followed by rapid growth, causes a supply-demand imbalance, which is why the sucrose accumulates in the leaf. High sucrose initiates that yellowing I spoke of, and really what that does is it accelerates aging and and senescence. The cause is any form of stress that significantly slows down growth and reduces that sugar sink size or strength. And a chemical option is being investigated as a management possibility. I'd like to thank our funding providers, Sugar Research Australia and QDAF, for their support over the last 6-7 years. And I'd also take this opportunity to acknowledge all the many researchers that have been involved in the YCS research in that time. And I make particular reference to Dr. Fricke Botha, who was managing the entire YCS program from the beginning up until November last year, and was instrumental in coordinating and what we've learned about YCS to date. And the many technicians who work tirelessly in the field, and a big thank you to productivity services and sugar services, SRA adoption staff, and the many growers who assisted in trials and sampling, and anyone else whom I've missed. Sorry, there's so many people, I probably have missed someone, who have helped us along the way. Thank you very much.
Thanks, Gerard. We've got time for some questions now. If anyone would like to either put them in the chat box or unmute your microphone. I have a comment here from Matt Hessian. The 2016 harvest in Gordon Vale was also heavily influenced by a very almost no flowering, and the crop continued to grow throughout the season. Also a very low CCS season.
Yes, so no flowering, but also low CCS. Yep, Matt, all I can say there to you is that there are many issues surrounding CCS, as you know, and remember that YCS only affects a small part of the stalk for that period of time. It's not affecting the crop from the very beginning. You don't see yellowing from the moment it comes out of the ground to the time it's harvested. And so when you send your stalks to the mill at harvest time, it's CCS for the entire stalk. So all I can say is that our data shows that when you look at the period of YCS impact and you test for sucrose in the stalks at that time, particularly in the internodes directly associated with the symptomatic leaves, there is no CCS penalty.
Okay, thanks, Gerard. At this stage, we've got one more question. This is from Joanne Michael. Do you think that the insects could act by affecting growth?
Good question.
The last question.
It's a difficult question to answer, because that's why we're conducting the insecticide trials now. But it is possible if you have a really high, I think it's possible if you have a really high insect pressure and it can take carbon away from the leaf and stop it getting to the stalk, then that will reduce the growth, yes. Okay, and if it impacts in that way and reduces growth, then you have the potential for that sucrose to accumulate in the leaf and for the leaf to go yellow. Insects are tricky things, and I'm not experienced enough to comment on it, and Gordon, Kevin Powell would be able to, but sometimes, you know, they can inject things into the plant and disrupt signaling between the leaf and the stalk and try and keep that sucrose there for themselves to feed on. But we haven't found any evidence of that, and it's still early days, and I know Kevin wants to do a lot more work in that space. So yes, potentially they can, but I think, you know, insects are one part of the story. We know that if you slow the crop down by many other means, you can still induce YCS, but we just really want to know the significance of insects and its role, or their role in YCS.
Thanks, Gerard. We have a question from Bryce. In the Burdekin, we purposely stress our seed cane sources by reducing irrigation and nitrogen rates. The aim is to reduce internode length and prevent lodging. These seed cane sources don't seem to show high rates of YCS. They do show stress, as you would expect, in the form of yellowing, leaf curling, and reduced growth, but not YCS.
Yeah, that's a good question, Bryce. I'm not sure I have an answer. I've seen YCS in the vertic and in the field, and actually I've been to some of those trials, and I would say to you as well that I've been into about 3 years back. I took samples from one of Catherine's propagation trials there, just near the Brandon station, and I did check it for sucrose and starch, and the symptoms and the pattern was right. So I have seen YCS in those trials, in those plots, and I have confirmed it. But you're right, it doesn't always correlate in all of those fields. So you have to have all the symptoms, all the planets align, I would say, to cause that tipping over. If you don't get that sucrose building past that 200, you're not going to see the yellow. But that doesn't mean that it's not sitting at around about 180 or 190. It just hasn't pushed past that tolerance level for whatever reason. Maybe there's not as much photosynthesis happening to produce as much sugar for whatever reason, in terms of if you have less water, a little less nutrition, then the plant lacks a little less vigor as well. And so photosynthetic rates may be down, and therefore you don't produce enough sugar to push it past the upper tolerance level. That could be the reason. But I have seen it in some of those fields, and I have confirmed it.
Thanks, Gerard. We've got a question from SRA's Nathalie Piperidis. As climate change is happening worldwide, is YCS happening in other countries?
Good question, Natalie. I haven't personally travelled overseas to look at any of this, but I know people who have, and they say they see it. But when you talk to the folk, the international folk, they say, no, we don't have YCS in our country. But Australians who go over and look at it say, yeah, well, I would call that YCS. Now, I haven't personally been. I think it would be visible in other crops, in sugarcane in other countries. It's just that they tend to call it just natural senescence. And really, when you think about it, YCS is induced senescence. It's bringing on aging that would normally happen, but it's just bringing it on a lot faster. And once you get rid of the cause in terms of that slow growth rate, once it gets growing again, then it grows out of it and it disappears. It's just that it induces it. So good question. I think it does exist overseas, but once again, as I say, I haven't personally been over to look at it, but I know people who think that they have seen it when they have been visiting.
Hey, thanks, Gerald. But because your tongue slipped a little bit, I was just having a following question. So does it happen in other crop, that same kind of stress things in Australia?
I don't know of it happening in any other crops. You can induce it in other crops, like there's been plenty of studies done, like David Braun has done it in, induced it, similar things in maize with his mutants. You can certainly induce it in corn, and it looks exactly the same, but I don't know of it naturally occurring in any other crops in Australia.
And Salam has made a comment there as well to say that similar symptoms occur in Papua New Guinea and Indonesia.
I might comment on that. Do you hear me?
Yes.
Yes, I can hear you.
Yeah, well, in New Guinea, they call it golden leaf syndrome. It might be a similar thing, it might be different, but it's more spectacular, I'd say. In Indonesia, I've seen symptoms in Indonesia that I would call YCS, but they just say it's stress, but it's typical symptoms, actually, of YCS. In China,
Yes.
I heard that they have it, but I haven't seen it.
Okay.
Yeah.
No, that's good. Thank you, Salam. You're right. They call it golden leaf syndrome in PNG, and we did actually look at some samples of that. and there are lots of similarities, but they're at a genetic level, at a gene expression level, there are differences, but it's hard to get good samples to bring back for those types of analyses when you're trying to collect samples in the field and get them into liquid nitrogen and dry ice to shut everything down to preserve the RNA. But yeah, that's good to know. And you've travelled extensively, so in your experience because of your involvement in YCS from the very beginning as well. So thank you.
A question here from Matt Hessian. What was the YCS situation this year? Was there YCS reported in all districts? And on that topic, Rob Miller has replied and said, we had pretty significant YCS in the Burdekin, almost as severe as we have seen it.
Yep. Thanks, Matt and Rob. YCS was very slow to get going this year everywhere. We had no early reports in December. There was the odd bit here and there, and I questioned whether it was actually YCS or not. But yeah, we didn't see YCS until more recently, and I know in the Burdekin there was a big wave of it, and we were monitoring it in Herbert, and there were some patches there, and so we did get reports, but it was a lot later this year, definitely, and it hasn't been as bad as a whole across the December to April period as it has been in other years. In fact, the last few years haven't been so bad. I talked to the Burdekin region now because recently got to see a particular field there that had YCS in it, but it had a lot of other things going on, and it puzzled us. But the soil and leaf nutrient tests have come back and shown that it's low in potassium. So it had slow growth rate for the last 3 months or so, and there's been yellowing getting worse and worse. But in that case, we now know that there's been a nutrient deficiency that's been instrumental in causing the slow growth rate. And of course, in that situation, we've seen YCS combined with nutrient deficiency symptoms.
Okay, a couple of comments in the chat area. Kevin Powell said, I saw YCS-like symptoms in Papua New Guinea this year. Serge has said YCS in Kamala to Inniston in the Plain Creek area for the last couple of years has severely impacted yield, and especially CCS, with cane being condemned. Claire Gersh has said ongoing YCS south of Serena this year. And I have a couple more questions, Gerard, if you can bear with us. From Stuart, has any correlation between farming system, controlled traffic, and YCS incidents for given environmental conditions?
Yeah, wording, but you could type it in.
No, I haven't been involved in any of that, but I would suggest that controlled traffic is very important, because if you can reduce compaction, and while studies on the roots showed there was no difference between YCS and controls, that's true, but we do know that compaction has a serious impact on the size of the root system, and of course the root is a very big sink, there's no doubt about it. So if you reduce the size of the sink there, then potentially you increase your risk of YCS. So look, I can't comment on farming practice because it's not an area that I've been involved with, but I think that all of those things are important, and that if you've got YCS in a particular, if you've got YCS in a particular field and you keep getting it in that field every year, then you've got to have a look at what potentially might be going on in there. And so, if you can, go and start off and look at the soil health for starters. Then yeah, maybe look at farming practices and things that see if you can identify what's different about that particular field and why it keeps getting YCS. Look, YCS appears in a field this year and doesn't in the same field the following year. It stops at a headland. It doesn't go across a headland. You know, what disease does that? It's not a disease. You know, it's something that you've got to identify. If you can identify what's causing stress in that field, what has an impact on growth rate, then you'll be able to risk manage YCS.
Okay, we've got time for one more question. I've got a question here from Peter. Gerard, did the 3 stalks you displayed, were there internode length differences come from the same field? If so, they would have grown in similar environmental conditions. So why did the internode lengths vary?
Good question. Those 3 stalks came from different parts of the field. And what I would suggest there is that in those parts of the field, there was something completely different going on. That you'll notice there that in those photographs that the length of the stalks were different, but also the thickness of the stalks were different. So there was a lot going on there. There was not good farming practice going on there. I would suggest it had serious nutritional deficiencies, and there was differences in the soil types. So there was fair range within that field that could cause those discrepancies. And so you need to look at both length and girth and look at the total volume, because the 3rd stalk, the shortest one in there, it had the same length as the stalk on the right, the one that had the largest internodes, but the lengths were the same, but very different in differences in thickness. So the shortest length was very thin, even though it had shortest stalk was very thin, even though it had quite long internodes. Its volume was still very small.
Okay, thanks, Gerard. We might wrap it up there. Thank you all for attending today's webinar. We hope that you found it of value and are keen to get your feedback. In the chat box is a link to a survey that we'd like you to complete. It will only take a few minutes of your time and it will allow us to improve our webinars in the future. I've also included a link in the chat box to a YCS information sheet that you might find helpful. So thank you very much to Gerard Scalia and to Phil Ross as well for his introduction. And we look forward to talking to you all shortly. Thank you.
Thanks, everyone.